My name is Caroline Sarbieski. I'm a member of the Imaging, Evaluation, and Treatment, or I-EAT Swallowing Laboratory here at Purdue University. Our lab studies swallowing throughout the lifespan. In this project, myself and my co-authors have investigated differences in swallowing physiology between young and old adults. Swallowing is critical to survival, wellness, and social interaction. It is also a complex action involving many muscles and nerves in the head and neck. We know that even with typical aging, changes happen to these structures and the swallow mechanism. It is critical to document these changes and find ranges of performance in both young and old healthy adults in order to use this information to compare against populations with swallowing disorders. Further, in addition to changes, variation can be seen in swallowing event sequencing, or the pattern of gestures that need to happen in order to protect the airway during swallowing. Certain patterns seem to be robust across ages, while others have not been extensively investigated. Our first aim was to determine the order of swallowing events in young and old healthy adults in order to reinforce or supplement previously researched data. Our second aim was to examine duration and reaction time differences in the swallow of these two groups in an attempt to replicate prior well-designed studies. Participants included 8 younger adults, 19 to 23 years of age, and 8 older adults, 65 to 79 years of age. A video fluoroscopic study, or real-time X-ray study, was used to evaluate swallowing. Each participant performed 3 trials for 2 consistencies, 10 ml of thin liquid and 5 cc of pudding. Table 1 depicts events or gestures of the swallow that occur or contribute to the protection of the airway established from previous research. One term that I will define is bolus, which is the material, whether it be food or liquid, in our mouth after we have manipulated it and it is ready to be swallowed. Some of these events are known to happen in a specific order, and for others the exact sequence has not been established. The second table depicts the sequencing patterns that have been found to be robust, partially supported, or in need of further investigation, highlighted in different colors in accordance with those 3 categories. The third table depicts duration and reaction time measures. Duration measures describe when a bolus passes from one anatomical structure to another, and reaction time refers to the time it takes for an action to happen in response to the bolus passing a certain anatomical structure. Percent agreement was used to descriptively compare previously established rules in the literature to events found in analysis. We used a Wilcoxon rank sum test to compare the duration and reaction time measures between younger and older healthy adults given the small sample size at this time. Regarding AIM 1, we found that laryngeal elevation always occurred before upper esophageal sphincter, or UES, opening, and UES opening always occurred before hyolaryngeal approximation across both age groups. Also, UES opening occurred before or with bolus at the UES, 95% of time across groups. Maximum opening of UES occurred before maximum pharyngeal constriction 93% of the time. When investigating the last rule that is under investigation, laryngeal elevation before or with bolus past mandible occurred in 88% of swallow trials in younger adults and in 71% of trials for older adults. Regarding AIM 2, descriptively pharyngeal reaction time, pharyngeal transit time, and laryngeal vestibule closure reaction time were found to be longer in older adults. However, these differences were only significant for pharyngeal transit time for thin liquids, seen on Figure 2. Aligning with previous studies, some patterns of sequencing, such as laryngeal elevation before UES opening and UES opening before hyolaryngeal approximation, were found to be robust in swallowing event sequencing across ages in our sample as well. The pattern of laryngeal elevation before or with bolus past mandible was found less often in older adults, which could reflect reduced airway protection in older adults due to aging. Increased PTT, or pharyngeal transit time, in older adults has been shown by several prior studies as well, and likely reflects reduced sensory motor responses in typical aging. In future studies, increasing the sample size may help us validate some of these preliminary findings. Thank you to the I-EAT team for their contributions, as well to an internal grant provided by the SLHS department that helped with data collection.